Okay friends, so now we are going to talk about another important topic which is called ultrastructures of Archaea, Methanococcus, Eubacteria, E. coli and unicellular eukaryotes. So if you remember the earlier discussion in the earlier lectures on ultrastructure and connection of the classification or characterization of these features of bacteria which are required or responsible for keeping these microorganisms into different classes, different phyla and different categories. So basically it is the extension of the same to understand more detail about it, individual level and different at the level of bacteria, fungus and other parasites. So we will first talk about the ultrastructure. Means like a complete microscopic structures which can give you the inside or in and out of the particular microorganisms, right from cell wall, membrane, internal organelle and all kind of structures. Now if we talk about the ultrastructures of the Archaea, that is the Methanococcus, Eubacteria, which comes like E. coli comes under the Eubacteria, and one of the unicellular eukaryotes. So we talk about both the three domains, that is Archaea, Eubacteria and Eukarya in this topic. So ultrastructures of Archaea, Eubacteria and unicellular eukaryotes. So let's talk about the features of these Archaea and Eubacteria first. Now what is most important here is because this is nothing to explain nothing to describe the detail to understand it, but I think this is just you have to remember these points, nothing to explain in this lecture, but to read and remember all these things. For example, cell type. So if you talk about the cell type of the features, you find in prokaryotes and in prokaryotic cells like Eubacteria, bacterial prokaryotic cells, you find the lack of membranous variations. So there's a difference in the membrane variation where you find because of these differences, the bacteria adapt the conditions, different conditions in the physiological responses they have, and the Archaea have a different conditions to respond to the environmental conditions, and that's how their habitat is different, their ecology is different. So cell wall is different from bacteria in Archaea. That lack of peptidoglycans in Archaea, it contains the pseudopeptidoglycan basically, proteins and polysaccharides, whereas in bacteria it contains a highly thick peptidoglycan in case of gram positive especially, and in case of gram negative also, along with the lipopolysaccharides. Now cell membrane lipids is different. In case of Archaea it is a ether linked, whereas in case of Eubacteria it is a ester linked lipid layer. So this ether and ester link also provide the strength to the bacterial populations to grow in the harsh conditions and in the mild conditions. Ribosome is different. 70s ribosomes in Archaea, but protein compositions are different. 70s ribosome present in bacteria, but the bacterial proteins are different from the archaeal proteins. Genetic material, single circular DNA, histone like proteins present, single circular DNA, but no histone except in some bacteria. Polyamines are present, which is not present in Archaea. RNA polymerases, multiple type of RNA polymerases are present, which is complex, just like in eukaryotes, whereas in case of bacteria, it is only one type of simple RNA polymerase of multiple units are present, like alpha, beta prime and sigma. Then we have reproduction. In case of Archaea, we have asexual binary reproduction, which is fission budding, whereas in case of simple fission bacteria, asexual type of reproduction present in the bacteria. Pathogenicity is generally Archaea are non-pathogen, where the bacteria are pathogenic. Some of the bacteria they may not have pathogens, but majority of them they are pathogens, which are especially the mesophiles. Habitats of Archaea are the extreme condition of environment: hot springs, salt lakes, deep sea, and anaerobic environment. Whereas in case of Eubacteria, they are ubiquitous, and soil, water are present, and they're present in the inside and organisms, some host system, because they are pathogens. Now metabolism, many are extremophiles, methanogens, halophiles and thermophiles, whereas Majority of the bacteria, they are the mesophiles, and some of them, they are photoheterotrophic, so they form their own food, like cyanobacteria, and chemotrophic, they need some chemicals to produce their foods. Examples, for example, Methanobrevibacter, Methanobrevibacters, and Halobacterium sulfolobus. These are the examples of the genera of the Archaea, which have a number of species in each genera. Whereas in case of bacteria, you have E. coli, Streptococcus, then we have Bacillus, Cyanobacteria, especially which are photosynthetic bacteria. Now, each of them we will discuss about the methanogens. Methanogens are methanobacterials and methanococcal, methanomicrobial, then we have methanosarcinales and methanopyrales. So, these are some orders of the methanogen bacteria, which has a number of genera in each order. Now, so what is most important here is the productions of methane, as we discussed in the last lectures. Last step is anaerobic degradation of organic matter. So basically, they anaerobically they degrade the organic material. So usually they live in kind of a sewage water, sometimes some sewer lines, where they find these organic materials, organic matters to decompose and to form the methane gas through these organic carbon matter. Now, ultrastructure of Archaea, that is Methanococcus. Archaea cells develop the plasma membrane, external layers, extra layers, then we have a cell wall, then S layers, and then we have capsules and slim layers. Then we have archaeal plasma membranes, lipid made up of hydrocarbons derived from isoprene units and 5 carbon branch molecules. So this is the chemistry behind the Methanococcus, where the membrane compositions is defined, how the membranes is composed, how the layers of the membranes are present, are formed, and these chemical compositions of this membrane make the membrane more rigid, more resistant to various harsh conditions of a temperature and several other conditions. Now, comparison of the archaeal and the bacterial membrane. In earlier lectures, we also talked about in brief, but here you can look into the comparison. In case of archaeal membrane, we have monolayers, right, and bilayers. So bacterial membranes, ester linkage is present. See, this is called ester linkage. Whereas in case of archaeal membrane, you have the ether linkage. Now, this ether linkage is a strong linkage which makes the membrane more strong and more protective. So this is the chemistry behind the these two membranes. Now, this bond is important to understand CH2OC ketone bond, right, CO bond, whereas this is the aliphatic group, carbon, and here it is only, there is no ketone group, it is only CH2, not CHR. So, this is the fatty acid chain is present. Again, this is basically head of the group in both the cases, but this bond, which is called ether bond, is basically responsible for the strengthening of the membrane. So this is the basic difference between these two membrane. Now, then again we have a Methanococcus cell wall. If you look into, we have a S layer, then plasma membrane, S layer, plasma membrane, and cytoplasm, right? Then S layer means like it is a glycoproteins are proteins of 20 to 40 nanometer thickness. So this basically layer provide the thickness and the strength to the cell wall on the outer cell wall of the this archaeal bacteria. Now, the cytoplasm of archaeobacteria are similar to the bacteria, and they contain the inclusion bodies, polyhydroxy alkanoate. These polyhydroxy alkanoates, basically polyphosphates and granules, glycogen granules and gas vacuoles, they are present in these cytosols. Ribosomes are present, nucleoids are present. Nucleoids are basically the regions where the chromosome is present, single stranded chromosome present, and sometimes the plasmid molecules may also present, just like a bacteria, but it's a very, very rare. The cytoskeleton proteins which are present in this bacteria, archaeal bacteria, are the FTS. This FTS is a tubulin homologs. This tubulin homologs means like role in the cell wall division. So they can have a proper division of a cell wall at the time of division, cell division, or the sexual reproductions, or the binary fission, or budding. MREB, which is actin homolog. This actin homolog of eukaryotes, it's rod shapes. Then you have a crenatin. Crenactin is basically actin like unique to Crenocata. This Archaeota is a bacteria class for the Archaebacteria, which is basically characterized by the crenactin, and this crenactin is basically the tough protein which provides the strength to the cell. Now, if you look into detail of the ribosomes, same sedimentation coefficient of 70s ribosome with the 50 and 30s ribosome small and subunit, whereas the 16s rRNA, 23s rRNA and 5s rRNA in some Archaea also have 5.8s rRNA like eukaryotes. So they are more close to the eukaryote. I mean, they are just like a closest to the between bacteria to eukaryotes. Proteins 68 more than bacteria. Bacteria has a less number of proteins. They are group 1, universal, Archaea specific, and then shared with the eukaryotes. So many proteins are also shared with the eukaryotes also. The key features of these ribosomes are resistant to the antibiotics that inhibit bacterial ribosomes. So many bacterial ribosomes are resistant. So the antibiotics which are inhibiting the protein synthesis, like a tetracycline, chlorphenicol, they may not having affecting the translation blockade process in case of Archaebacteria because of the resistance in these ribosomes. Now, nucleoids are the chromosome containing regions, are localized region where the chromosomes are present, which is circular double-stranded DNA, often polyploids. It means not N, it is 2N, 3N, 4N. Compaction proteins which provide the compactness to this, and like HU-like proteins which is in bacteria, then alba proteins which is like a Crenacta, which is Crenacata. Crenacata is again the Archaea group of bacteria, and alba proteins are present in the Crenocata. Histone-like a protein which is present in the Crenocata, which is close to the eukaryotes. So Urea-Chiota is basically slightly close to the eukaryotes, early primitive eukaryotes, because they have some histone-like proteins are close to the histone-like protein, which stabilizes the DNA molecule in the nucleoid region. Condensin is also present, which helps in the chromosome segregation at the time of cell division, both in budding and fission. Now, the surface appendages are basically surface appendages like pili, then cannulae, then you have a hemi. So pili is made up of a type 4 pilin homologs. Then we have central humans, unlike bacterial pili, because bacterial pili is different from the archaeal pili. Function is adhesion. In case of bacterial pili, it also adhesion, as well as it also provide a role of transfer of the genetic material through horizontal gene transfer during the conjugation process. Cannulae is pyrodictyum class, and then it is hollow tubes connecting daughter cells and networks, different cells during divisions, and then function is not yet known. Hemi is a grappling hook-like structure, and they help to attach the cell surface on the biofilm. So suppose the biofilm formation, so they help in like hook-like structure, they help in the surface, and they catch hold of surface on the this. So flagella is Archaea. As we discussed the flagella about the bacteria flagella, we have both Archaea and bacteria flagella. They have like motor-like structures. We have a tubulin, and we have like a flagellin and spiral filamentous flagellin, which help in the motility of the bacteria and swimming of the bacteria. The superficially similar to the bacterial flagella, but slightly different in terms of like a thinness. This is thinner than the bacterial flagella, 10 to 30 nanometer versus 20 nanometer in case of bacteria. And it's not hollow. Multiple flagellin types are present. Then hook-like structures, which is hard to distinguish from the filaments, and no true basal body. Just like in bacteria, we have a true basal bodies present, which we call them as a motor, which help in rotating the flagella and to move the bacteria forward or backward movement depending upon the flagellar movement. Assembly of these flagella growth at base like fourth pili, not like bacterial flagella, and energy source is ATP hydrolysis, not protein motive forces. So we have a energy driven process is also present in bacteria, which is called ATP driven hydrolysis, which lead to energy production for the movement of the flagella to move forward or backward movement depending upon the situation. And it helps in the rotation based repulsion or the rotation based propulsion, like it keep moving by rotating, it move ahead, it rotating, it move ahead. And then similarly it Otherwise, if anti-clockwise or clockwise movement, make it forward and backward direction. It can also go to the reverse direction, that is backward, but no run on the tumbler mover, right. This is the characteristic feature of the flagella. Now, Eubacteria, example of E. coli. Protobacteria is one of the group. Then besides Protobacteria, there are many other class of group of bacteria present in Eubacteria, but E. coli is belong to this Protobacteria, and we call them as Eubacteria. This is the prokaryotic organism by the features, because no nucleus, true nucleus with a membrane bound, no confined chromosomes bound with the nucleus, and the membranes is also of a different kind. Organelles are not non-membranous. Now, the cell wall usually contain the peptidoglycan, we call murine, both in gram positive and gram negative. Gram positive, you have a thick peptidoglycan, in gram negative, you have a thin peptidoglycan. Now, cell membrane has ester linkage, unlike the ether linkage in case of Archaea, and 70S ribosomes present with the protein synthesis, with a different protein than the archaeal bacteria. The genetic material is a single circular DNA, no histone protein for stabilization. It is a circular DNA, double structure, single circular DNA, and reproduced by binary fission, we call asexual reproduction. Can be autotrophic, that is a photosynthetic, chemosynthetic, or heterotrophic, like cyanobacteria, which requires some chlorophylls and to some chemoautotrophics and so on. Now, it found the diverse habitats, soil, water, inside host, parasites and so on. Some are the pathogenic, like E. coli, Streptococcus, Mycobacterium tuberculosis and so on. Others are beneficial, like nitrogen fixation bacteria, then fermentation bacteria, antibiotic producing bacteria, they are all good bacteria. Now, the E. coli is usually found in the human intestine, and indicator of the fecal contamination. So, sometimes this E. coli because of the anatomical structure of the organ, right, for urine and for feces, it might come out and may cause the urinary tract infection. We call UTI. So, E. coli is the most common, E. coli is most common bacteria causing UTI infection, for the same reason that they present in the human intestine, and it comes through feces, and it may come in contact with the from the urine, through urine. The indicators of the fecal contamination, if the fecal contamination is more, coliform will be more. So, you can check the coliform, number of coliform to check the fecal contamination. It may cause urinary tract infection, traveler's diarrhea and serious food-borne diseases. The ultrastructure of Eubacteria, as in Archaea bacteria, the peptidoglycan was present, but we have different structures, different bond, different strength as compared to the Archaea. Here we have different shape, and for the protection the cell wall is present in the form of murine. Plasma membrane is a phospholipid bilayers with the ester linkage fatty acid, unlike ether linkage. Cytoplasm, which lack the membrane bound organelles, contain the enzymes, metabolites. Then we have a nucleoids, single circular DNA molecules. It is not enclosed by the membrane, unlike the Eubacteria, right. Plasmids are basically, unlike the eukaryotes. Plasmids are also present, plasmids are very commonly present in the Eubacteria. So, there was extra chromosomal material, acquired several gene which help in horizontal gene transfer, transmission gene from one bacteria to other bacteria. So, this is called a genetic transfer, or the word we call horizontal gene transfer. Then inclusions or storage granules are present, glycogen, polyphosphate, sulfur, gas, vacuoles. Then you have a flagella, pili, right, locomotion, flagella and attachment, conjugations, pili fimbriae are present. Capsules, then we have a slim layers present in some bacteria. It provides the protection against the host. So, basically these are the defense mechanisms of the bacteria which help in protecting bacteria from not engulfment, not phagocytosis of the immune system, and once they escape out from immune system, then immune system may not get any kind of infection. So no infection establishment, no infection cause if the immune system suppress the production of this. Then you have endospores. In some genera, example Bacillus, Clostridium, highly resistant resting structures present. So these endospores producing bacteria are those bacteria which produce endosperm, and these endosperm may cause the resistance, and this resistance is basically make the bacteria more resistant to survive in the harsh conditions. Moreover, the heat, the sterilization conditions, heating conditions or high temperature conditions may not destroy this bacteria because spores remain there. So once you grow the cells and you even kill the cells, spores remain unkilled or undestroyed, and when the spores are undestroyed, then these spores can re-emerge, right? And after re-emergence of these spores, again the spores reappears and you call. So as far as the ultrastructure is concerned, so in a summary of this lecture, the ultrastructures of the bacteria, that is Eubacteria, Archaea, right, is important to understand, to look into the variation among the ultrastructures. Why ultrastructure is important? Because in ultrastructure you can identify from outer cell membrane to cell wall, plasma membrane, to cell organelles, right, and their whole mechanistic approach of the synthesis of protein, replication and all in a single domain. Now comparing the two different kind of structure, ultrastructure, you can identify the responses of this category of the cells, that is Eubacteria and Archaea, in different environment, in different conditions. Their physiology responses are different. When the physiological responses are different in these two categories based on the different ultrastructures as we just studied, it means these two organisms may have a different habitat, right? They have a different function. They have a different targets to kill. For example, if you want to kill the Archaea bacteria by heating, it will not kill, because the ultrastructure tells that their membrane is ether bond, a strong membrane, and because of this it will not destroyed by high temperature. Similarly, if you increase the temperature and you kill the cells through high temperature, then the Eubacteria will be killed because they have a peptidoglycan, lipid protein layers, and this get dissolved, this get fragile, and it cause the loss of cells through lysis during the heat process. So similarly, this is one example. Similarly, different physiological conditions based on the different ultrastructures we studied, the responses are different, and these responses in the environment give you different signals, different conditions, different growth rates, right? And so accordingly you decide the pathogenicity. You can think of some target to kill these bacteria under different condition based on your requirement. So the importance of the studying ultrastructure is to know how about the inside of these microorganisms, how these microorganisms are being made. Until unless you understand the whole ultrastructure, you cannot control, you cannot manage these microorganisms, right? In order to properly manage and control these microorganisms, you have to study the ultrastructures and to know what these ultrastructures is telling you about the complete intactness of these microorganisms. Okay. With this, I think we close this lecture today, and we have another lecture soon next. Thank you very much.